Module 4, Bioinformatics. In Module 3, we covered a wide array of techniques and methods for capturing omics data, from dense phenotyping to the breadth of metabolites and microbes in our bodies. As in Module 3, where it would be impossible to describe every type of omics technology, in Module 4, we'll provide a general survey of the many ways the dense data sets generated by these technologies are housed, organized, and queried. The goal of this module is to give you access to tools you can use to answer questions about genetic and genomic data that you might encounter. In particular, more and more companies are generating gene-based dietary programs, and many people are interested in these tools. We'll focus on these gene-based types of dietary intervention strategies more deeply in Modules 11 and 12. This module will include an overview of the more user-friendly databases for querying information about genes and their function. And this video will focus on a database called GeneCards. To navigate to the GeneCards database, go to www.genecards.org. And it's a good idea if you have two screens, to follow along in this video as we walk through the steps of using the GeneCards database. So here we are in the GeneCards homepage, and I have to say that GeneCards was created in 1997 by the Weizmann Institute, and I've actually been using this database of databases ever since grad school. So it's an amazing place to start when you want to query, find information about a gene. It has links to just about every major database that you can think of to, as it says here, interrogate genomic, transcriptomic, proteomic, genetic, clinical, and functional information. So it's a really great resource to start with. So on the home page, the very first thing it says is explore a gene. So let's think about a gene that you might be familiar with. We're going to talk about, we're going to choose glucokinase. I can write the word glucokinase and I can query it, or if I know the gene symbol, I can query directly to the gene symbol. And here it is. The gene symbol is GCK, and it takes me to the gene card for glucokinase. But I can also, if I'm not sure of which gene I want to look at, or if I want to look at possibly a whole pathway, I could type in the word glucose. It will give me an error saying there's no symbol for glucose, but then I can tell it to go ahead and search for the word glucose. And now it's bringing up every gene that's been associated with glucose or glucose metabolism. And these are called mini cards. And this can be really useful as well because it's sorted in the order of the top hits, so the ones most strongly associated with a search term. And that gives me a whole list of genes that might be important to me or might be interesting to me as I query the database later. So we chose glucokinase, and here it is. That's the mini card. And again, it's going to take us directly to the card itself. I will tell you also that there are many ways to navigate around in GeneCards. And if I know, for example, that I just want to look at the expression, I can click on these words up here and I can go straight to the part of the card that talks about expression. I'm not going to do that right now because I want to walk you through all the information that's provided in a GeneCard. But I just want to show you that there's lots and lots of ways to navigate through the GeneCard database. So the very first thing that it's giving me here is aliases, are all the ways that this particular gene has been referred to, primarily glucokinase, but it's also been called hexokinase 4. So those are good things to know in case you're reading an old paper that refers to the gene in some other way. Then the next section is giving you a summary, and it's telling you what does this gene do, what's it involved in. And the nice thing is that not only does it give you information from GeneCards itself, but if there are other types of summaries, it gives you links to link out to those other places. So for example, here's the entree summary, and I can click on that link, and it takes me to the entree database, which is part of NCBI. And here's a little summary of what the gene does and some links within the entree database. The nice thing about GeneCards too is that every time you link out, it opens a new window. So navigating back to the original place you were in GeneCards is super easy. And here's, if you like the really kind of basic information, it takes you even to places like GeneWiki, which is where genes are described in Wikipedia, which everybody loves Wikipedia. It's always an easy source to look at. So I'm going to go back to GeneCards. The next section is called Genomics, and it's telling you a little bit about the general structure and location in the genome of the gene. So the first little section here is listing the known promoter and enhancer sequences. And I can click on these, I can click on any of these links, and it can take me to the different databases that describe promoter and enhancer sequences, and actually give me the sequence and the location of the promoters for this gene. I'm not going to do that now in the interest of time, but if you wanted to know more about the specific sequence of a promoter or enhancer for a particular gene, you can click there. Here it tells you a lot about where the gene is located in the genome. So in this particular case, glucose kinase is on chromosome 7. It starts at position 44,144,271, and it goes from 44,198 to 170. It tells you here that the size of the gene, the total gene, is about 54,000 bases. So it also tells you down here that I can click here to go out to the UCSC database and click on a custom track that's been created in that database that links to the GeneCards database. In terms of its chromosomal location, if you'll remember, I think in an earlier video, you remember that the small arm of a chromosome is called the P arm, and the long arm is called a Q arm. And these different markings on the chromosome result from the fact that when you stain a chromosome with Gemson, it produces these bands or stripes. And so scientists long before you or me have labeled the different bands, and that provides some location for different places in the genome. So this particular gene is located at 7p13, so on the long arm of, on the short arm of chromosome 7 at location p13. And again, I can click out to the HGNC database. That's the Human Genetic Database, the Entree database, or the Ensemble database, which you've already seen in an earlier video. And it will take me directly to the sequence and some other information about the genomic structure of the gene. The next section is called Proteins. I can click here, and it will link me directly to the UniProt SwissProt database, which is the largest database of proteins. This is a great database as well. It tells me about the structure. It tells me about the catalytic elements in this particular protein, where it functions and what processes it's involved in. So lots of information in UniProt, SwissProt. And again, there are other databases of resources that can tell you a little bit more about the actual amino acid sequences and the structure of the protein. Weizmann Institute, when they created this database, it became super useful, and after a while, they kind of were running out of money to keep it current. So what they did was they very, very wisely partnered with biotech companies to provide funds to keep the database current, at the same time providing information about assays and products that the different vendors are selling that are related to the genes of interest. And actually, this is kind of great, because if I want to do an assay on glucokinase, I can go straight to GeneCards and look up all the different sources of finding antibodies, for example, which is just a really nice and handy thing to do. So it was a really smart thing to get vendors to contribute to keeping this database current, at the same time providing kind of a unified source of where you could get information and products for your laboratory research. The next section is called Domains and Families. It tells you a little bit about what families glucokinase might be found in. So you can see it's related to hexokinase, which of course you already knew. It tells you a little bit. The next section is called function. So it's telling you about the primary functions of the particular gene you're querying. And here is a section, a table called phenotypes. So phenotypes associated, which are sometimes pathologies, but often just bodily functions associated with this particular gene. So things like, of course, fasting blood glucose, body mass index indirectly through its effects on fasting blood glucose, HbA1c, birth weight, type 2 diabetes mellitus, and you guys knew about that. It tells you about the ontology. So this gene ontology tells you about the domains of the protein that are actually similar to other kinds of proteins. And then here it's telling you about phenotypes. So there's a neat little table that tells you about some of the phenotypes that have been associated with this gene. So in this case, there's some these are almost always pathologies in this particular table. Abnormal C-peptide, abnormal heart morphology, abnormalities of the autonomic nervous system. And you can, again, these are all clickable out to the human phenotype database. So this is really kind of pretty cool. That's a pretty cool database too. So if I click on abnormal C-peptides, it takes me out to the human phenotype database. It'll take me, of course, directly to the thing I just looked up, but I can look up all kinds of phenotypes in the phenotype database and see, you know, what's associated with a particular phenotype. I can go here. Again, these are some products that I can use to query. This tells me about the localization of the gene, and again, you can see that from, that figure is coming from Uniprot. And again, it tells you about ontology and other things that are found in those particular parts of the cell. In this section, it's called pathways and interactions. So it tells you which kinds of pathways the gene is associated with. In this case, galactose metabolism, glucose metabolism, and I can click on these links and it will take me directly to the pathway databases. Here, it gives me a little picture of the interacting genes with glucokinase here in red. I can look at these string figures, which are often really interesting to look at to see how the genes are directly or indirectly related to one another. And I can right-click on almost any graphic in this particular database and copy the image and paste it in another document. Here, it's giving me interacting proteins, and I can click on those and get some more information about those proteins and processes. In this particular section, for drugs and compounds, these are either drugs or compounds that are used to query or analyze the protein, or drugs or compounds that have been developed with this protein as a base. In this case, these are compounds that I'm probably going to use to test the activity of glucokinase. Things like D-glucose and glucosamine and dextrose can be used in assays looking at function. The next section is called transcripts, and again, this is going to allow me to link directly out to various transcript databases. If I click here on the RefSeq number, it's going to take me directly to the NCBI database. And here I've clicked on the transcript, so it's giving me the mRNA sequence. So while you saw that the gene itself was about 54,000 bases, I'm just going to scroll down sort of quick. This is telling you that the gene itself goes from, in this particular reference, from base 1 to base 2,757. So clearly lots of intronic and other types of sequence that gets removed as this gene gets processed during gene expression. And it gives me the features and tells me where things start and stop. And if I scroll all the way to the bottom, it gives me here the nucleotide sequence of the coding region of this gene. And that is in NCBI. But there's actually several places where I can link out to coding sequence, not just NCBI. I can also link to Ensemble and Entree and other databases as well. It's also giving me some products that I can use to query and to ascertain gene expression. In this case, it's telling me a little bit about the structure of the gene itself and whether or not there's any alternative splicing in that gene. The next section is called expression, and it's actually E expression. What the how these graphics are generated is from, in this case, RNA-seq or microarray or SAGE databases, providing a compilation of all of the places that this gene has been recorded to be expressed. So you might see that the gene is expressed or at low levels, or at least it shows up on microarrays and other places. And this is the kind of the compendium of those data. But if you scroll down a little further, it's telling you this is an actual query of where the protein is found. And here it tells you, of course, that it's found in liver, which is where it does its main job, as you guys know. And it's also found in the fetal heart, where glucose metabolism is a little bit different than the mature organism. Again, giving you some more links out to other kinds of expression databases. The next section is called orthologs, so genes that are found in other organisms that either are exactly the same gene or very similar, perform similar functions in different organisms. And here it's listing mouse, chicken, lizard, African clawed frog, and the zebrafish. But you can actually look there, it says see all 40, and it will expand that table and show you all of the different organisms that have a glucokinase gene. Paralogs are the proteins in the human genome that are similar in function and structure, and of course hexokinase would be one of those. In terms of variants, this is actually a section that links out to dbSNP and provides a list of all the variants in this gene. You'll see here it's showing you 5 of the variants, but if you look closely it says see all 10,819 variants. And I could click right there. That is actually clickable, and it will open this table up, and it takes a little bit of time to open that many records, but I can get this right in here in this reference. It tells me about, I can click and go directly out to dbSNPs, which I'm going to do in just a second to show you the record that's found there. It tells me the type of variant, and then it tells me over here, in this case, this is a variant in which at this position in the genome, you might find a C, A, G, or T, so it's highly variable. And it also says that those variants at that particular place are coding sequence variants. They're called missense. They cause missense of the protein or a premature stop codon. So if I want to look more closely at that particular SNP, I can click on it, the link. Again, it takes me, that particular reference takes me directly to dbSNP, which is the NCBI database of all known SNPs. This is a really big database. So here's the one I just clicked on. Here's the name of it. It tells me what gene it's found in, glucokinase, and dbSNP gives me all kinds of information about where it is, what the variant is. It also gives me location, and dbSNP tells me all of the references that have reported this particular SNP, and so that's really important too. It tells me about the sequence surrounding the SNP, and this is useful for if I want to create some kind of assay that queries that particular SNP. I can take that sequence right there. And it also tells me what populations the SNP is found in and what the frequency is of that particular variant. So that's all of the dbSNP information. I'm going to go back to GeneCards. And so in this variant section, it's giving me some structural variants. So these are things like copy number, or in this case, these are all copy number variants: tandem duplications, gains, insertions, losses of sequence. So these are structural, which you guys heard about in an earlier module. This section talks about disorders known to be associated with this particular gene. So when there's defects in glucokinase that's been associated with hyperinsulinemic hypoglycemia, maturity onset diabetes of the young, diabetes mellitus, monogenic diabetes, and maturity onset of the young type 2. So this is a really neat little database called GeneCards, which is actually part of GeneCards that you can go directly to if you're interested in some defects. So I'm going to show that to you in just a second. And then at the very bottom here is a list of publications. So it's listing the top 5, or maybe the most recent 5, or the most important 5. But you see here it says see all 746. And if I click on that link, it's going to open up a list of all the references that are currently in PubMed for referencing out to this particular, or referencing or mentioning this particular gene. I'm going to go, and then down here are all the partners and some of the links that you can go to get directly to these vendor sites. I'm going to scroll really quickly back up to the top and show you that some of these other databases are clickable directly. GeneCards is kind of a cool database because if you're particularly interested in a disease, they're not all listed here in GeneCards, but for example, say it's the summer's coming up and I'm interested in skin cancer. So it's bringing up some options for skin cancer, and I can say go, and it will come and it will bring up again mini cards that are related to every outcome listed in the GeneCards database that's associated with skin cancer. And at the very top is squamous cell carcinoma, which is skin cancer. And then I can link directly to in this card information about that particular disease. So that is very useful when you're querying something particular. A friend of yours is ill, or a client or a patient, and you want to know more about their particular disease. Here is a kind of a neat little graphic that shows you how squamous cell carcinoma is related to many other types of cancer. So I'm going to go back here to the GeneCards database. Just as a conclusion, so there's some other databases again that you can click on up here. Any of the sections that I showed you, I can click directly to the section here. I mentioned expression. I can go straight to expression without having to scroll through the entire page. And as you see, this little section remains at the top. So if I wanted to look up CRISPR assays, I can go directly to CRISPR assays and not have to scroll through and looking for it. So pretty useful database for you in the future. If you're answering questions about genes or genetics, this is a great place to start.